Hello, everyone. Today I will be presenting my final project for Mediterranean food and culture. And so my project centers around the neuroscience of food preferences. Within my project, there's 5 distinct sections, beginning with the introduction that touches on why it is imperative to study this matter. Then we move on to a distinction I make between taste and flavor, which is a very important distinction for this topic. Thirdly, we talk about taste variation, and in particular, how this is mediated by our genetics. Then we have the progression of flavor preference, and I utilize 3 different timescales to explain this topic. And then lastly, we have the conclusion, which I present some of the key takeaways from this research and from the findings. So for our introduction, we want to touch on why do humans have variable preferences for food, as this is really what this topic is centered around. And to do so, we really have to begin with the omnivore's dilemma. As we have touched previously in class, the omnivore's dilemma describes the fact that us as humans are really unique as a species because we have a very well adapted digestive system. Our digestion system can process an array of different types of plants and animals. And thus, because we can process so many plants and animals, we have to make a choice of what we end up ingesting. This is a choice that is really unique to our species in comparison to others, because other species have a lot less or a lot more biologically constricted digestive systems that limit them to a certain set of nutrients. So for us, when we come to this dilemma, as you could say, we are presented with the choice of what to ingest. Well, we often can end up making the wrong choice for our nutritional health. And that is why it's important to understand this matter of why we make the choices that we do when selecting our food. So as we move on, we have this map that displays the different types of food across the world. And this really begins to show that because of the grand variety of food we can process, we end up making very distinct choices at a global level, but also at the individual level. This particularly represents different foods across different cultures, and we can see an array of different ingredients being utilized and different plates being preferred over others in different regions. But as we want to also understand how this happens at the individual level, we can move on to the lens that we will be looking at it through. We want to understand this food preference through more of a scientific, rigorous lens, and that's why I pose the nature versus nurture debate. This debate is very common to lots of biological and scientific studies and research areas, particularly because it is a recurring theme throughout our research. So what this the debate really expresses is the fact that our behaviors can either be genetically determined, which would be nature, and these are how they occur because of innate differences we have in genetic variations from person to person, or whether our actions and behaviors are modulated by our environment, upbringing, and life experiences, as is seen here on the nurture side. And so, within the lens of food preferences, we want to understand that is whether our genetic variation that we have as individuals that leads us and constrains us to make certain food preferences, and that is what causes such deviation for these food preferences, or we want to see it's whether it's our environment and these different experiences and expectations we have about food and different properties about food that we learn about, or subconscious mechanisms taking place that affect our food preferences. And so, through my research, I would like to posit that our food preferences are primarily mandated by our expectations of flavor and our internal state at the time of feeding, but are confined within a set of genetic constraints and thresholds. And so that is what really the argument I will be developing across this presentation. And to do so, we really have to start making a distinction between taste and flavor because they're often used interchangeably for the same property, while they really do not represent the same thing. So first, we ask ourselves, what is taste? Well, for taste, I would say a rigorous definition of it would say that it is the perception produced or stimulated when a substance chemically reacts with the taste receptors cells located on taste buds in the papillae of the oral cavity, primarily on the tongue, but there's also taste receptors within the stomach and intestines. So now that we have defined that, we want to talk about these 5 properties that we can actually end up tasting. And these 5 properties include sweet, sour, salty, bitter, and umami. Sweet foods would be like, as we know, ice cream. Sour could be something like lemon that has also a very acidic property. We would have salty, which would be maybe some Lay's potato chips. Bitter, which would be commonly something like coffee. And lastly, umami, which is more of the obscure one, but we could think of it as some very savory things like meats and also, say, soy sauce. And so we ask ourselves, how is it that we go from ingesting this food to then actually perceiving one of these 5 tastes? So this is the basic taste pathway that I would like to express to you guys, and with this lovely diagram. So firstly, we begin here with the food particle, which would be the food that is masticated within the mouth by your teeth. And once we have this breakdown, which is also aided chemically through the saliva, there is certain, let's say, substances within the food that are actually picked up by these taste receptors. So these taste receptors, once they have binded these certain substances, have an electrochemical transduction of the receptor signal to then relate to a nerve. This nerve has an action potential, or we could just say impulse, that travels and carries neuronal information to the gustatory cortex, which is right here at the top of our temporal lobe. And so once we have this, we can actually, that we get a cognition of the detection of this particular taste that is being processed. So we ask ourselves, how is it that this happens at a deeper level? Well, I would like to express first the salty receptor and kind of go into the ins and outs of how the neuroscience behind this works, because I find it very interesting. So firstly, we would have to distinguish the fact that the salty receptor for this tastant, which is salt, is actually an ion channel. And this describes the type of receptor that it is. And so after mastication of a salty food, what we're actually detecting is the Na plus sodium particle. And so that is what actually enters through a taste pore. And these taste pores are actually at the top of our taste buds within the papillae of the oral cavity, as actually we can see here. We see where this taste pore is at the end of the single taste bud. So after we have the sodium entering the taste pore, this enters through passive diffusion because there's more sodium on the outside and thus goes into a place of less sodium concentration. And upon entering, what happens is it raises the membrane potential of this. And we don't have to get too much into the electrochemistry of it, but let's just say that the more sodium that enters, it raises the membrane potential. And as the membrane potential opens up and increases, we get opening of more sodium channels, which are called voltage-gated sodium channels. And this causes an influx of sodium within and starts a process that we know as calcium-mediated exocytosis. And this, in a nutshell, is really just calcium is prompted to enter. There are some complex mechanisms that occur that causes the fusion of these vesicles to the outer membrane. And the vesicles are the circular things that have little dots within them. And after we have fusion, we have release of a neurotransmitter, which is in this case serotonin. And this serotonin then binds to a gustatory afferent neuron, which is the neuron that we see here, or they'll say the nerve. And so after we have that, it binds to other receptors and we get the neuronal impulse. And we don't actually have to touch on too much on the details of that, but let's just say that this is really a prime example of chemical to electrical transduction, as we have a literal chemical, which is sodium, and that is complexly converted to a neuronal electrical signal that then travels to our brain for it to be detected. So as we move on, similarly, we have a sour receptor. It really follows the same mechanism as the salt receptor. However, it is actually the hydrogen plus ion that is what is being entered. And so this is this acidic proton is actually present in all of these sour compounds like lemon, and as we have expressed other things. So as it enters, once again, we have passive diffusion. We allow for more hydrogen influx. And after we get that, we get, like, once again, calcium mediated exocytosis, and then we release serotonin, same as previously, travels as an electrical impulse in the nerve and is picked up by the brain. Then we have bitter, sweet, and umami receptors, and these are actually a different kind of receptor. These are not ion channels that have a direct entering of the chemical, which in previous cases was sodium and hydrogen. And this time we actually have a binding of the bitter, sweet, or umami chemicals to what we call a G protein here. And once we have this binding, there's a chemical transduction, which really just activates what we call a G protein, leads to calcium release, then a sodium influx, and activates what we call an ATP channel, which then activates the afferent neuron here, which travels to the primary gustatory cortex. And so There's a lot of details within the neuroscience of how this works. I find it particularly interesting, but really what we have to take away here is the big message, which is really the theme that we have a chemical to electrical transduction. The chemical is being converted to a neuronal impulse, and this is what we get as really a rigorous perception of a taste in. As when you taste something, you're like, okay, this is sour. This is really how it occurs. It's quite fascinating. So now, once we get to the primary gustatory cortex, we want to talk about the intensity of this activation and of this perception in particular. So over here to the right, we have a diagram that reveals that it's quite a simple property, but it's hard to study. But what it's revealing is that the more you have of a substance, the more the neuron is activated and the more you get of that perception. And that is what research has shown. The more salt that's in your food, the more the salt receptor is going to be activated, the more neuronal impulse, the stronger the intensity of the neuronal impulse, and the stronger the perception of salt. And over here to the left, we have a very interesting graph that really just proves or reveals what we were talking about earlier, that we're limited to these 5 different tastes. And this really, it only shows 4, but what this is showing is that each taste receptor is particularly modulated to respond optimally to one taste in, and that is why we have these 5 different types of taste receptors. As we see here, sweet optimally responds to sucrose, NaCl optimally activates the salty receptor, and so on. So now that we've touched on taste, we want to make this physiological distinction to flavor, which is flavor not only is the taste, which would be these 5 taste sense, but it takes it a step further and is really what we more commonly think of as taste. Flavor describes the attribute of a substance derived from the perception of its taste, odor, temperature, irritation, and texture within the mouth. And so then this shows that other senses and cognitions have been shown to have an impact on flavor perception. So an example to really show this is, say you take 2 jelly beans. We have a cherry jelly bean and we have a strawberry jelly bean. If you're to ingest both of these normally, you will be able to make the flavor distinction of cherry and strawberry. However, we have to differentiate this from the taste or isolate the taste. If you were to plug your nose and you remove the odor component of this, then as we know that they share the same temperature, irritation, and texture, once you isolate that, if you plug your nose and ate the cherry and strawberry one separately with your nose plugged, you would actually only be able to taste the sweetness of it. You really wouldn't be able to distinguish the fact that is flavored as cherry versus strawberry, because this really comes from the odor and these different kinds of senses that are involved on our flavor perception. And so this is really particularly interesting because both of these are impacted separately by genetics and environment, and thus that's why we have to make the distinction. So moving forward, we're going to talk a little bit about taste variation, and this is really particularly how our genetics modulate our perception of taste sense, because this is really the main mechanism that is occurring here. We really have our taste. What I'm arguing is that it's really biologically constrained. It's really physiologically constrained. And the level of this taste perception is really limited by our variations in our genes. So to reveal this, there has been lots of research conducted on taste receptors, and previously the ones that we previously talked about. And so this is what we call receptor polymorphisms. And polymorphisms talks about the fact that a single nucleotide, or it can be multiple, but in this case, it's been shown that it's single nucleotide changes in our genetic code lead to different genotypes of this taste receptor. And so these different taste receptors process the taste sense in a differing manner, and thus we want to explore how this impacts our food preferences. So firstly, we have here some researchers did studies on the CD36 gene, and we found when we can either have the genotype A, B, or C. And so in a normal CD36, which modulates fat preference, we see a normal level of it. When we have lots of high CD36 expression, we get overstimulation, and thus we have low preference for fat. And then the converse is true, is that when we have low CD36, we have high fat preference, and this can often lead to a higher risk of obesity. As we move on, we have the TAS2R38 gene, which has the same 3 genotypes, and we see that this actually relates to tasters, non-tasters, and super tasters, but it's really within the same mechanism of processing of the genotypes. And this just shows, once again, that we have normal bitter preference, or we have high bitter preferences. You have low the genotype B, which is low bitter receptor activation or sensitivity, and then super tasters, which we have high sensitivity and thus increased aversion to bitter things like, for instance, alcohol. And then lastly, we have the TAS1R2 gene. And once again, we have the three genotypes. We talk about the same kind of thing. The sensitivity of sweet is modulated by these different genotypes and thus leads to a different preference for sugars and the sugar taste in. And this is particularly important within this discussion because here we're seeing a true genetic variation that leads to selection of food preferences. An individual that's a super taster is going to be more aversive to alcohol, whereas someone that's a non-taster might be more seeking of it because of the actual taste profile that's being activated. But this is all within a tasting threshold. So this is really talking about the sensitivity and how much these receptors are being activated. And so we really want to talk about how this is impacted by other biological functions and to further express the importance of this tasting threshold. So it's been revealed that hunger actually can modulate the sensitivity of receptors. When we're very hungry, we actually end up seeing an increased sensitivity to sugar, as researchers found, and a decreased sensitivity to bitterness, which was actually really interesting. And then on the other hand, when we have pregnancy, we have a decreased sensitivity towards sugar and an increased sensitivity towards bitterness. And this is modulated by hormonal changes. And then lastly, we also have repeated exposure. And we can see that if you eat the same food over and over again, if you don't like it, it won't particularly change your liking of it because this is something we'll touch on later. But it actually affects the receptor sensitivity, as some research has found, because the repeated exposure desensitizes some of the receptors to the tastins. And so, yeah, let's move on forward. So now that we have talked a little bit about this threshold and the sensitivity of tastins within the receptors, we want to talk about the implications of diminishing taste. And what I mean by this is the fact that as we grow, our taste sensitivity actually decreases. We get our taste buds actually die and turn over very quickly. But as we go older, this rate diminishes and thus we have less taste. So why is this important? Well, our food preferences as we age don't actually follow this biological process linearly, or this genetic process. Since we get less sensitivity, you would think that our food preferences would thus be shifted within the constraints of our own sensitivity to the five taste senses, and also would follow linearly with the fact that we would be seeking more intense taste since our sensitivity has decreased. However, our food preferences as we age and just in general are a lot more complex than this. So we want to really start asking ourselves, if it isn't the genetic and biological constraints that are solely determining this, then what other types of mechanisms are taking place to be affecting this? And so that's when we move on to the fourth section, which is the progression of flavor preference. And flavor preference lies, I think, at the intersection of our senses and psychology, as we have explained when we describe flavor. So I wanted to first describe 3 different time scales that this occurs in, and so we can really understand the food preferences, particularly the flavor changes in preference. So the first one we have is momentary changes in preference, and we have 4 different let's say environmental factors that are taking place, nurture factors that can affect our change in food preference. And these mechanisms fall outside of genetic variation, fall outside of the differences we have to our taste receptor sensitivities. So firstly, we have here this red plate, which represents the idea, which comes from gastrophysics, which was particularly, that researches exactly this matter. And really what they found was that if you present food on red plates, people are actually going to eat less because of the color associated with red and stop. You'll actually see a diminished food intake. So it's just an interesting impact on our food preference, and also the fact that we'll choose, we're less likely to choose foods plated on red plates. Further, we have what I find a really interesting study, which is they found that prices actually impact our food preference. If you're given 2 plates of exactly the same, say, mac and cheese, but you're told one is $100, another one is $10, and you try both of them, you're actually more prone to say that you like the $100 one, even though these 2 foods share the same chemical properties. And so this can't be because of genetic variation. This can't be because of the differing sensitivities of our receptors when 2 different people that have these sensitivities are still being swayed by the price tag to it's the same food. So this can occur through processes like cognitive dissonance, where you're justifying this increased purchase value or increased value with maybe a linear increase in food quality or taste. And then next we move on to music. It has been shown also that music and ambiance within a restaurant can actually impact how we perceive this food and what food we pick. So we'll actually make different choices if there's different genres of music being played. And also within, say, a genre of music being played, we can actually impact how we appraise the food's flavor and value. And next we move on to emotions, and our internal state can actually affect how we perceive foods. So within this particular study, they showed individuals happy faces or sad faces in the two different testing groups. And they showed that the happy faces, after showing these faces, they were made to drink the same orange juice. And they showed that when they showed the happy faces and thus increased positive emotion, people perceived the juice to be better and to be actually sweeter. But when we had sad faces shown, people perceived the juice to be less sweet, as with the negative emotion pairings. And so here we just see these momentary changes that fall outside of genetic variation and are very interesting. So as we move on, we see changes that occur, lifetime changes in preference that occur across a lifetime scale. And firstly, we have this picture, which depicts nostalgia. And so lots of studies have been conducted and shows that individuals tend to pick foods that remind them positive of their childhood. And so it's interesting because different individuals can have obviously different childhoods, and this will actually directly impact the different foods that they will try to consume or that they have a preference for through an emotional connection through them with it, which is a memory. And this is not prone to genetic variation. So this is very interesting. Secondly, we have cultural consistency. There's been lots of studies that show that individuals tend to prefer foods that are consistent within their own culture. And this is linked with social identity. As we begin to form this cultural identity, we are more prone to stay within it, as food is also part of culture. And thus, outside of the actual properties, chemical properties of these food, we're more likely to like them if they're culturally consistent. And then as we move on, we have this healthy salad that depicts actually our health concerns across a lifetime. And as we grow older, we become more health conscious in general. And as our health deteriorates, we really want to make these positive food choices. And so these healthy, positive food choices are subject to changes in preference, because as we seek healthier foods, we actually begin to rationalize our liking of these foods, even though the food property hasn't changed throughout our lifetime. So for instance, individuals who know that they need to stick to a lower salty diet or a diminished salt diet actually over time end up liking foods that are diminished in salt because of the health benefits that are associated with it, rather than actually the sensitivity to the salt within the food. And lastly, we want to touch on generational changes in preference. And in particular, we have these three. The first one depicts the French taste revolution, which is something we really touched on in class. And this depicts the fact that previously we liked foods that were a lot more artificial, flavors were mixed and heavily intense flavors. But as we know with Pierre the chef, he kind of revolutionized more the chef as a humble interpreter, where plates end up tasting like what they look like, no mixing of flavors, a little bit less intense of taste and spicing, more sticking to herbs. And really the takeaway message from this is that there's been little to no change in our biology. However, we end up choosing different foods over time over what we find to be socially acceptable and what is kind of mandated as the out cuisine, the highest level of cuisine. We end up gaining a liking towards this. So just as now we wouldn't like chicken with sugar as they did back then, this isn't really constrained within our biology, but rather a social component of generational change in preference. Then we move on to this picture of Italian American cuisine that we have here by the spaghetti on the American flag. And we talked about how when Italians came here to America, they actually saw an increase in meat consumption and satisfaction with their meat consumption. And this is because of the societal implication of eating meat, which displays wealth, and also the lack of availability of it in their home country. We saw this change in preference that occurred across generations. And this preference, the generations aren't having differences, say, in their umami receptors to make them like me anymore. It's really, once again, a social mechanism taking place. And lastly, we have this depiction of technology, and this talks about the different technological advancements that happen across generations and how these have actually been shown to have differences in food preferences. For instance, you could say Gen Zers, there's certain phenomenons like the foods that are trending within, say, let's say TikTok. During quarantine, there was the Gigi Hadid pasta, which was a pasta that was famously made on TikTok. And actually, we saw a number in the increase of its consumption and ordering at restaurants through solely the popularity of its media display, but not really through any biological mechanisms. And so different generations are prompted to different sorts of media and different expectations of food. And this can really have an impact on what we choose to eat. So lastly, let's conclude this conversation. And some of the key takeaways is that we do have some innate taste preferences and aversions, as we saw with the differences in receptor sensitivity. And also, I can add the fact that it's been shown that babies, have a natural preference for sugar, and we also have a natural aversion to bitterness because of the evolutionary implications of poison. And so these are true. We do have these genetic preferences. However, these really only begin to form a structure for within the flavored preferences to work in. They're not really the tell-all, be-all mechanism for our food choices. So as we move on, genetic variations, as I said, cannot solely explain our preference. And our environment, society, and internal state actually mediate our food selection across different timescales. And this is really the main mechanisms that our food preferences are working within. And this mechanism can particularly be seen as top-down processing, as how it affects our appraisal of food, because bottom down would be our genetic sensitivity to the stimulants, which would be the taste sense, and this would solely shape our appraisal of food. But it's actually the top-down processes, which are our appraisals, our expectations, what society thinks, the ambiance, these other things that are actually outside of the actual chemical properties of the food that are making the biggest impact on what we end up liking or not. And then I think this is a very important matter to study, because understanding where food preferences are derived from is imperative for public health and education and nutritional behavior, because if we can really understand why we make the choices we make, we can begin to make better choices. So thank you so much for listening, and I hope you guys have a great rest of your day.